Okay, seems like a good time to start. So we are a week out from our next exam. I'm hoping by tomorrow I give you a better idea how much information that will cover. I'm hoping to get through all this genetic stuff or come pretty close to maybe just finishing a few things up on Monday. We'll see where we're at. I will not get to our next chapter on viruses. So that chapter is available for you to see, and the homework is available. I've been trying to change the due date to that, and it doesn't seem to be changing when I tell it to, so I might have to contact Top Hat to work that out. But I'm for sure viruses will not be on our exam next week. So we've got enough as it is. So there will be a few more questions on this exam. So you might have to work a little bit more efficiently. Most of you had a lot of time last exam. Question. So we're going to be which chapters? Starting with the chapter 5 on the eukaryotes, chapter 7, the growth, 9, metabolism, and then 10 and 11 are the genetics. So it's a good bit more, the eukaryotic stuff and the growth stuff is it's a lot of definitions and terminology. Metabolism and genetics a little bit more process oriented, kind of things that are a little more abstract maybe. So yeah, so I would say, don't leave that homework till right at the end. Most of you have been good about that. I don't think it's a ton, but maybe, hey, let's get that, finished up over the weekend, and then give you some time to start going through things. So, all right, so let's jump in. Last time we were talking about Messes and Stahl experiment. So that's that most beautiful experiment in biology that's proved that DNA is replicated semi-conservatively. So think about what that means. One of the original strands is conserved, and then a new copy is made of the other strand. Okay, so if we get into a little bit more thinking more molecular in terms of how DNA is copied, and we call that replication. There's not actually a bunch of nucleotides, and remember what a nucleotide is. That's the nitrogenous base, the deoxyribose sugar, and a phosphate. That's a nucleotide. DNA is a big polymer composed of monomer nucleotides. But when you're piecing that DNA together in the cell, it's not a bunch of nucleotides actually that are floating around. It's actually a triphosphate nucleotide. So actually you have all these triphosphate deoxyribonucleotides floating around. So remember, those phosphates have those high energy phosphodiester bonds. So those nucleotides actually carry with them the energy for the polymerization reaction. And the terminology gets a little bit cumbersome here. We refer to these as nucleotides with the D in front of them. So that stands for instance, guanosine triphosphate deoxyribonucleotide. So the D is the deoxyribonucleotide. Again, it's a little bit cumbersome, but that just means, again, that it carries with them the energy for the polymerization reaction. So here's kind of a schematic of how that works. Remember, there's going to be another DNA strand, say right here, that's the template for which kind of nucleotide, which base needs to come in. This 3', this is the hydroxyl group. That's the polymerization reaction always extends at that 3' side. And here's a guanine triphosphate nucleotide here. Again, 2 phosphates are cleaved up. Why is it 2 instead of 1? Well, 2 makes it even more energetically favorable. This polymerization reaction happens really, really quickly. And then we've just got, again, a longer DNA strand here. Okay. All right. So some of the enzymes involved here, and remember, we're talking about prokaryotes. So remember, their chromosome is usually circular in prokaryotes. Where does replication begin? It begins at a site called the origin. So that's kind of intuitive, hopefully. Okay, the site of, we'll call it the initiation of replication. Okay, in prokaryotes, there's usually going to have just one origin. Okay, so in this little graphic here, I'm not crazy about this graphic because it's kind of hard to see the different colors, but the little blue triangle there, that's the origin, or sorry, rectangle. Green rectangle, sorry, that's the origin. Okay, so in prokaryotes, there's usually only one. And usually that's a site in the chromosome that's fairly rich in A, adenines and thymines. Because remember, when the adenines and thymines are complementary and they only have 2 hydrogen bonds rather than 3 with the G's and C's. So they break apart a little bit easier. Okay, and replication is going to proceed in both directions from that origin. So you can see here what we call a replication fork is opening up, going both ways. And the new strand, that's what I don't like about this here. The new strand is kind of a light purple, and the older strand is kind of a dark purple. I don't know why they don't make those colors more contrasting so you can see that. Sometimes this is referred to as a theta structure because it resembles the Greek theta. Helicase is the enzyme that is unzipping the DNA, or in other words, separating the DNA strands. I see that referred to sometimes as melting the DNA as well. It just means the 2 strands are coming apart. You need a special enzyme to do that called helicase. And then the other enzyme that makes sense to talk about right here is topoisomerase 4. And that comes into play when we've got the 2 rings, the 2, I guess, the daughter chromosome, and maybe you could say the original one, they actually stay linked together, kind of like 2 links in a chain. So you need an enzyme to come along to cut them apart and reseal the bonds again. So we call that a concatemer. Okay. So concatemers. Okay, so right here, this is considered a concatemer because, again, prokaryotic chromosomes, they stay linked. So the topoisomerase, again, cleaves that DNA, unattaches it, and seals that back up again. Okay. And again, this enzyme, topoisomerase 4, that's going to be unique to prokaryotes, because eukaryotes don't have circular chromosomes like that. Okay, so you end up with a new copy of the chromosome. And again, the cell is going to do this when it's getting ready to divide in half, binary fission. All right, just eukaryotes, because eukaryotes typically have so much more DNA, they're going to have more than one origin of replication. They're going to have multiple origins and replication forks, again, extending in each direction. If they only had one, it would just take too long to copy all that DNA. So all that's showing there. All right, so what are some more of the enzymes that are involved in this process of replication? The star enzyme, if you only remember one enzyme, remember DNA polymerase 3. Why do we call it DNA polymerase 3 if it's so important? Well, there's about at least 5 different DNA polymerases, and they were named by the order that they were discovered. So that's why it's DNA polymerase 3. But this is the enzyme that adds those triphosphate nucleotides to the growing strand. So here's the old, think about directionality for a minute here, because that's always kind of confusing. The old strand here is 3'. Okay, so the new strand, this is going to be a 5', and the new strand, like I have already said, is always going to be adding on to that. There's a 3' hydroxyl group there. Okay, so the new strand is always adding on to the 3' end. Okay, the DNA polymerase 3 has to work in that direction. So we call this the leading strand on the top, because it's adding nucleotides towards the replication fork here. So on the other strand, DNA polymerase still only adds onto the 3' end, so it's going to kind of add in fragments like that we're going to call Okazaki fragments, and I'll write that in a minute. But still overall going towards the replication fork, but it has to do it in a piece by piece way. Let's see, what else? So here you can see the helicase, that's the enzyme that's pulling the 2 strands apart, melting those hydrogen bonds between the bases. There's also some stabilizing proteins here that just helps keep that replication fork open. So a little bit more about this enzyme. Like I mentioned before, it polymerizes really, really quickly. It adds about 500 to 1,000 base pairs per second. So really fast. And additionally, it has some proofreading function. Proofreading exonuclease function. So that just means that the enzyme actually adds the new nucleotide, moves on to the next one, but also checks behind it to make sure that it added the base that's complementary to the old strand. And if accidentally put the wrong nucleotide in there, then it can cut it out and add the new one in. Okay, so not only does it move really fast, but it can check and make sure that it's put the right base in. All right, so let's look a little bit more closely at that leading strand, because that's a little simpler than the lagging strand. One thing about that enzyme, DNA polymerase 3, it can't start from nothing. We say that, I have this written in a minute, de novo. It doesn't begin de novo. It needs something, it needs a 3' hydroxyl group to grab a hold of. So another enzyme, primase, will come in and synthesize an RNA primer at the origin. Okay, so we see that right here. And now the DNA polymerase can start from that short sequence of RNA, usually only about 10 to 20 base pairs. Whoops, let me write that here. So the primer is RNA, and it's 10 to 20 base pairs. Okay, now that is going to be temporary, because you can't have just little bits of RNA within the DNA. So that's going to be cut out later, but that gets things started. And again, here it's showing a little better, adding those triphosphate nucleotides. And remember, this would be the 3' end here of the new leading strand there. Okay, so again, let's make sure we write this temporary. All right, so that's just, that's just gonna go towards the replication fork until all the DNA is copied, basically. The lagging strand, though, there's a little bit more involved with the lagging strand. We have to have a series of primers synthesized. So you can see those in red in this diagram here. There's one here and here and here. And then DNA polymerase 3 is going to synthesize DNA in short segments. And short, meaning somewhere around 1,000 base pairs. Okay, but we call these Okazaki fragments. Okay, that's because that RNA polymerase only works in one direction. Okay, and then a couple other enzymes have to come in and get rid of those primers. That's DNA polymerase 1. Okay, we'll get rid of the primers, those RNA primers. So you see that's labeled right here. That's the kind of orangey blob there. It's getting rid of the primer, filling in the complementary base pairs to the conserved strand. And then one more enzyme, ligase, is necessary to kind of seal up the gaps. Apparently, a phosphate and a phosphodiester bond has to be added in there so that you've got the new strand as being one. So here's that DNA ligase right there. Okay, so on the leading strand, I think that's what this kind of blob is there that you can't really make out. That's going to be DNA polymerase 1 working there to get rid of the primer, and then of course ligase filling in the gaps. So that only has to happen once for the leading strand, but the lagging strand has to happen over and over again. Okay, so this YouTube video walks through that. It's only a couple minutes long. If you're kind of a visual learner and you like to see how that all comes together, that's kind of right at this level that I'm teaching it with the same enzymes involved and everything. So you're going to want to know what all those enzymes do: primase, DNA polymerase 3 and 1, ligase. Those are kind of the most important enzymes in replication. Just a couple other things to mention about replication. One problem that the cell encounters is supercoiling. So I'll come back to this in a second. I didn't post this, but if you have two strands of something that are wound together and you're trying to separate them, okay, eventually you get this basically twisting up ahead of where you're trying to separate them, and eventually you can't separate them anymore. Okay, we call that supercoiling. So that's a problem. The cell has to have some way to relieve that supercoiling. Otherwise, again, the mechanical stress of it doesn't allow the replication fork to open anymore. So the answer here, at least in prokaryotes, we call the enzyme gyrase. I think the textbook also refers to it as topoisomerase 2. But I always call it DNA gyrase. We can DNA gyrase. Okay, so that's working upstream here of the replication fork. That enzyme does a lot. It makes cuts in both strands of DNA, untwists it, and then seals the DNA back up again. Okay, so we can spend a lot of time on what that enzyme does, but it does a lot. And this is an enzyme it's a little bit different than the enzyme that does that in eukaryotes. So we'll talk about a few weeks from now some antibiotics that target that particular enzyme. So again, anything that only bacteria have is going to be a good target for antibiotics because it's not going to cause side effects to us. And also just going to be aware that there is some methylation that takes place. So a methyl group, CH3, is usually added right on to those adenine bases a few minutes after that new strand is copied. So there are slight differences in bacterial DNA, say from our DNA. Our DNA gets methylated too, but it's usually a different base. Usually it's cysteines. So slight differences there. What function does that methylation have? Apparently it helps define where the origin should be. So, you know, when those enzymes kind of know where to attach to start that replication process. We'll see in a little while, probably Friday, how it's involved in DNA and a particular kind of DNA repair mechanism. And there may be some regulation involved, meaning, you know, certain genes only get made into RNA when there's a certain amount of methylation. So kind of various functions. They're enzymes that add those methyl groups. All right. Okay, so replication is really a little bit simpler, at least in bacteria, because you kind of start in one place and then all the DNA is replicated. When you're thinking about how RNA is made from DNA, call that process transcription, not all the DNA is going to be made into RNA. So you have to have a particular place where it starts and a particular place mechanism for it to stop. So it gets a little bit more complicated. We will spend most of our time really thinking particularly about how DNA is made into messenger RNA, but whatever kind of RNA is made, whether it's tRNA or ribosomal RNA or primers even, we call that process transcription, no matter what kind it is. Okay, so, all right, so three events. We're gonna talk about it in terms of initiation. So, and brought in a lot of graphics that are not in your book. Your book tends to like talk about it a lot, and I don't think it gives enough pictures. So additional pictures than what are in your book. And the initiation is going to involve the main enzyme, RNA polymerase, and how it binds to the DNA to start copying it. Elongation, fairly, just kind of keeps on going. We won't spend a lot of time there. And then termination, there are a couple different methods that we're going to talk about how that happens. Okay, so you have to kind of keep those events straight in your mind and try to keep them separate from replication and then, of course, translation that we'll talk about next. All right, I have found this diagram useful. So in transcription, remember, only one of the strands of DNA is going to get made into RNA. So there's some terminology around which strand that is. But here's a DNA strand. The main enzyme that's working here is called RNA polymerase. In eukaryotes, they have a different RNA polymerase depending on what kind of RNA is being transcribed. But prokaryotes, they just have one, no matter what kind of RNA they're making, it's RNA polymerase. And that's moving down the DNA strand, adding on RNA, triphosphate RNA, ribonucleotides onto that 3' end. Okay, just kind of same direction as DNA replication. So like I said, DNA is 2 strands. Which strand is being used? Well, the strand that is used, we have several names for. We can call it the antisense strand. We could also call it the negative sense strand. And that should make sense because messenger RNA is always going to be positive sense RNA. We say if it's readable at the ribosomes, then it's positive sense. Okay, so it makes sense that the positive is always going to be copied from the negative. Okay. How does the RNA polymerase know which strand to transcribe? It knows. It knows somehow, and then we base our names based, we call it, oh, this is a set. You can't tell by looking at it, in other words, that, oh yeah, it's supposed to transcribe this strand and not that strand. Okay. I think it helps if we write in some nucleotides here. So let's see if the DNA strand here is TACGG. Okay, the opposite DNA strand up here then would be ATGCC. And of course, if that opposite strand was transcribed, it would be going in this direction. But let's say then the RNA, let's write in the RNA bases here, AUGCC. Okay, so hopefully you notice here the RNA strand, it looks the same as the other strand of DNA. Okay, so we call this strand the plus sense strand, or another name for that is the coding strand. Okay, coding because this sequence is going to be the same as the RNA here, except of course we've got U's instead of T's in the RNA. Okay, this strand that is transcribed is also referred to as the template strand. Okay, so hopefully that makes sense. If you're gonna, I was just making up an invitation to a party the other day, and I Googled, oh, templates for invitation. Okay, it's something that's already started. I just had to fill in where it was, and it was easy. Okay, so it's something you might use a template for a resume, for example. Okay, so this is kind of what's used to base the RNA off of. And then the opposite strand is the same code as what that RNA would be. So we call that the coding strand. Okay, that terminology, again, gets a little bit confusing. I certainly will admit that. Okay. All right, so let's move on. Let's compare that main enzyme in transcription to the main enzyme that we use for replication. Okay, they kind of sound alike. RNA polymerase, DNA polymerase 3. We talked about how DNA polymerase 3 needed helicase to unzip the two strands of DNA. RNA polymerase is able to unzip the DNA itself. It's part of its function. It doesn't need another enzyme moving ahead of it to do that. And no primers are necessary. Okay, so it can recognize the DNA and basically start making RNA straight from that. We've got uracil instead of thymine. Of course, an RNA. The proofreading, I talked about that, but I didn't assign a number. That DNA polymerase 3, we think it only makes an error about 1 out of 10 billion times that it doesn't fix. Okay, so that's pretty good, impressive fidelity. The proofreading for RNA polymerase is a little bit less efficient. Makes a few more errors. Probably about 1 out of 10,000 base pair does it make an error. Okay, but that's okay that it's not quite as good. The DNA is the blueprint. And if there's a mistake in that, every RNA that's ever made from that, there's going to be a mistake in it. So that's a big problem. Okay, if you write your recipe down wrong, every time you make those cookies or whatever, you're going to make it wrong. So that's a problem. But if every once in a while you make a mistake, oh, I thought that said a cup of salt instead of a tablespoon of salt, it's going to turn out bad, but the next time you'll probably read it right. So it's not usually a huge problem. All right, so let's talk about how transcription begins. So first of all, orient yourself. Of course, it's going to go off of that DNA template. And that RNA polymerase is going to attach to the DNA at a segment a little bit upstream of where transcription begins. So here's where transcription begins. Upstream of that, okay, we call this the promoter region of the DNA. Okay, so that's the promoter is where the RNA polymerase binds. And one principle to remember here, the more tightly the RNA polymerase binds to that promoter region, the more likely it will go ahead then and transcribe the gene. Okay, so there are lots of different things that will influence how tightly that RNA polymerase binds. There's something called sigma factor. It's a lowercase sigma there. Okay, sigma factor binds to the RNA polymerase and helps it recognize the promoter. Okay, and that's its only job. Once its RNA polymerase actually starts transcribing the gene, the sigma factor will fall off. Other regions that are important, there's this -10 region and -35 base pair region that we think are especially important for binding of the RNA polymerase. So -10 base pairs and -35 base pair. Again, those regions are important for RNA polymerase binding. And that -10 region in eukaryotes, we call that the Tata box. So you might have heard that before. In prokaryotes, it has another name called the Pribnow box. I'm not going to write that down. I decided let's just call it the -10 region because your book doesn't call it that. It calls it the Tata box, whether it's eukaryotes or prokaryotes. So I said, let's just call it -10. And trying to figure out if your book is wrong or whether there's some changing terminology. So we'll just call it the -10 region and that -35 region. Again, the more closely RNA polymerase recognizes that sequence of base pairs, the more likely the gene is going to be transcribed. This looks confusing, but to me, this diagram has helped me make some connections. So I want to point out a few things from this. This is sequences of the promoter regions. Let's see, I'll write this here. Sequences of promoters in E. coli. Some of them. Okay. And of course, the promoter, that's part of operons. So operons, let me just define that. A group of genes under the control of a single promoter. Okay, there's actually 600 to 700 different operons that have been identified in E. coli. Okay, you probably talked about one, the lac operon, and we're going to talk about that more hopefully Friday. But so just if you just look at that lac operon, remember, sometimes you forget what a promoter is. It's just a sequence of bases. Okay, and again, don't worry about what we call that. But the -10 region and the -35 region are highlighted because it turns out that when they have sequenced all these different operons of E. coli, they have found a consensus sequence. And that means a sequence that most of the time what commonly those base pairs are. Okay, so for example, 69% of the time, that first base is thymine. 79% of the time, the second one is thymine. So we call these a consensus sequence, and it turns out the closer the -35 and -10 base pair region is to the consensus sequence, then the more likely the DNA is transcribed. The more likely the RNA polymerase will recognize it and then transcribe the gene. Okay, so as to whether that DNA gets transcribed, it depends on, there's actually different sigma factors that can bind to the RNA polymerase that makes it bind more or less tightly. How well it recognizes that -10 and -35 sequence. Okay, all those play into, whether the gene gets transcribed or not. So there's a little level of regulation built right into the sequence there. Okay. All right, so that basically initiation, the binding of the RNA polymerase to the DNA. Elongation of the RNA is just the RNA polymerase here. That's that big orange circle. That's the enzyme moving down the DNA, making this
strand of RNA. It's adding on to the 3' hydroxyl group of that new RNA, following the template strands. Here it just says template and non-template. Remember, we called this coding, we called this negative sense, so that the RNA is going to be positive sense. Okay. And the RNA polymerase is melting those strands apart as it goes. All right, so that's elongation. Keep in mind that multiple messenger RNAs can be made from that same transcript all at once. You got multiple RNA polymerases moving down that strand. If it's information for a protein that the cell needs a lot of, it can make a lot of it really quickly. And also keep in mind where this is taking place. It's going to be, transcription is going to be taking place wherever the DNA is. So in prokaryotes, it's going to be taking place in the cytoplasm. If you want to be more specific, you could say the nucleoid region of the cytoplasm. And of course, in eukaryotes, this is going to be taking place in the nucleus, again, because that's where the DNA is. All right. How is transcription finished? Okay, we call that process termination. There's two methods. We think that probably they happen with about equal frequency. It's a little bit more complicated than that, but there's a lot that we don't really understand about transcription. So here's the basic idea of it. Sometimes it can depend on this enzyme or this protein. I guess it's an enzyme called Rho. This is actually a helicase enzyme.
Okay, and you remember what helicase does, it breaks those bonds between the base pairs on opposite strands. Basically, this enzyme moves along the RNA, and when the RNA is transcribed, it kind of has hydrogen bonding with the complementary bases that are very temporary, obviously, until it disassociates. But that Rho protein moves along the strand and breaks those hydrogen bonds and basically dissociates the RNA polymerase from the DNA and the new RNA strand. Okay, so one method is it involves this Rho protein. The other method is called Rho independent. So it doesn't involve Rho protein. We also refer to it as being sequence dependent, or sometimes it's called self-termination. So it's confusing. We got many different names for the same thing. But the idea is that there are built into the sequence of DNA some palindrome sequences. What's palindrome? Like the name Hannah is a palindrome. It's the same forwards and backwards, whichever way you read it. When we talk about palindromes in the DNA sequence, we are referring to, like, here's an example, GCCG. Okay, it's the same forward and back, but here's its complementary CGGC. Okay, so one is a palindrome of another because they complement each other. So that RNA strand that has these palindromes in it will kind of curve back around and make these hairpin loops that cause this mechanical stress and cause it to kind of loosen and fall away from the DNA strand. And there's usually a sequence of A's and U's, which, remember, only have 2 hydrogen bonds, so that dissociates a little bit easier. Okay, so mechanical stress causes that, we'll just say, transcription to end. There's a lot of details that we don't really understand here, and it gets a lot more complicated when you look deeper into this process. So just kind of the basic idea of how we think that happens. Okay. All right, so differences. And a few important differences to highlight between prokaryotic transcription and eukaryotic transcription. Besides where they physically take place in the cell, also recognize that in eukaryotes, everything is a lot more complicated. They have certain transcription factors like elongation factors, different proteins that have to be guiding the whole process that prokaryotes don't have. I mentioned that in eukaryotes, they have a different RNA polymerase for whichever kind of RNA is being transcribed. And in addition, eukaryotic transcripts are usually monocistronic. Cistron is from the Greek gene, so one gene, meaning we get, for each gene, you get one protein. In other words, each messenger RNA will encode for one protein. In prokaryotes, that's not always the case. The transcripts are often, or they can be, polycistronic. Okay, so many genes, meaning each messenger RNA may encode for a few different proteins. So that's operons, where we have, several different proteins, the production of several different proteins under the control of one promoter region. So it's a very kind of efficient way to regulate proteins that are necessary for the same thing. Additionally, in eukaryotes, there's some processing that takes place. So in order to stabilize that DNA, because it actually is going to have to travel from the nucleus out, to stabilize that RNA, messenger RNA that has to travel from the nucleus out to the ribosomes, there's some polyadenylation on the 3' end of the messenger RNA, and a guanine cap, actually modified guanine bases, added to the 5' end of the messenger RNA. Okay. And again, that's because it has to travel from the nucleus out to the ribosomes in the cytoplasm. Okay, and if you didn't cover up those ends a little bit, they're going to be subject to nucleases inside in the cytoplasm that would kind of eat away at that transcript. Also, that messenger RNA that's first transcribed from DNA, I actually call it pre-messenger RNA because there's some parts of it that have to be cut out. Okay, there are introns. You could think about those as intervening sequences that are cut, and the exons or expressed genes are going to remain. Okay, so it's a little bit of cutting that has to happen to get a messenger RNA transcript. And this graphic shows both those things quite nicely. Of course, here's the template DNA strand. A pre-messenger RNA is going to have both exons and introns in it. You can see that 5' cap. That's actually in eukaryotes. Make sure eukaryotes, that's going to line up just right at the ribosome, that 5' cap, and this polyadenine tail that protects the ends. A complex of proteins and enzymes called a spliceosome complex goes down and gets rid of those introns so that you only have exons left. Okay, and now it leaves the nucleus and travels out to the ribosomes. Okay, so again, that's eukaryotes. We don't have to have any kinds of splicing in bacteria. Just to mention, sometimes that splicing can be done in alternative ways in which you would actually get end up with a different product, a different sequence of amino acids. So if here the exons are squares and the introns are just little lines, if you just got rid of the introns, you would expect, you know, a messenger RNA like this, which would translate into maybe that protein. Maybe it's spliced a little differently and this yellow box is spliced out, you would get this. If you had a partial on the third one here, you would get slightly different. So again, it's another method of control to get slightly perhaps different proteins from the same transcript. Sometimes that happens in error. So we know some diseases like Duchenne's muscular dystrophy happens due to errors in splicing, suspected that maybe Parkinson's as well. So sometimes that happens on purpose, but sometimes things go wrong. Okay, we are just a minute from being out of time. I think we will start there and see how much we get through on Wednesday. Translation, regulation, a little bit of mutation, hopefully. So have a good day.